me. Now I'm in my slides. So I'm going to present a brief overview of Biodata PT, and would like to start with a little bit of history. So we started in someone's mind in 2010 with the first proposal for a Portuguese node of Elixir, and by then this discussion was all over Europe, because as you can see, only in 2014, I guess, Portugal joins the Elixir contract agreement, and this is when the Elixir was founded. In 2016, there is the foundation of the Portuguese node of Elixir, and the Biodata project starts at 2017. So we started with a single idea of a Portuguese node of Elixir to bring the Portuguese infrastructure of biological data to Portugal, but then it evolved between two paths, and these two paths were led by several institutions. The leads were INESC-ID and the IGC, but they were seconded by IBET, CCMAR, ITQB, and further on we have been gathering others. So in 2018 we resumed again merging the Biodata and Elixir, so becoming a single consortium, and later on by suggestion of the FCT and with agreement of several of the partners of the first consortium, we became a private, a non-for-profit private association. This is who we are now. Our vision is to support the transformation of Portugal in a country with the ability to conduct research and innovation with large volumes of biological data, and that Portuguese organizations will be able to translate this data into innovation for research, products, and services. And our mission is, of course, to make that possible by organizing and adding value to biological data generated by Portuguese research, enhancing and adding value through a national platform of biological information to different scientific domains. Also, computing resources, bioinformatics, and data management service, as well as training, will be available to capacitate these platforms' users. So by the end of 2021, our association, and this is all about people, but people belong to 12 organizations that you can see here, and these come from all over Portugal, and we are an open organization, so we welcome other organizations to join us. As I started by saying, the first idea was to found the Portuguese node of Elixir. So from day zero, we are aligned with the European space, and Elixir is the bioinformatics infrastructure, the European bioinformatics infrastructure. It is a landmark of the European strategy forum for research infrastructures, and it focuses on several domains as well of life sciences, namely environment, bioindustries, agriculture, medicine, amongst others. So what do we do here at Biodata PT as a Portuguese node of Elixir? So we will then maintain the Biodata platform of biological information with the following services. So we provide computing and bioinformatics resources, we provide data management services, we provide training in bioinformatics and data management. And we have an organics that is very similar to many other associations, and would just like to bring your attention for the communities, platforms, and services that are our most organic structures where all the operations occur. So communities and focus groups are domain-specific research communities and identify gaps in computing resources, analytical tools, and data management. You can see here several that are present in the Elixir. For instance, we will hear today about the plant sciences, but we also have 3D bioinformatics, human data, marine metagenomics, that is now the so-called microbiome community, but also microbial biotechnology. And our idea is always, and every time it is possible, to overlap with Elixir to take the most benefit and the state-of-the-art knowledge in these topics. As platforms support these communities to fill the identified gaps by creating new services and training units. In Portugal, we have an active compute, training, and interoperability data platforms. The purpose of communities, focus groups, and platforms, and I'm sharing this with you to invite you to have a look in our website and also at the Elixir website. And if you are interested, ask us for a link that we are sharing with our associates to invite people for our communities. So these groups bring together domain-specific experts across the national nodes. They capture the user needs into formal requirements, improve interaction and contact with user, drive the development of services, and this can be standards, tools, workflows, demonstrate broad impact of the infrastructure contribution to the users, help Elixir Portugal to demonstrate that the national services are global leaders. In addition, they will increasingly be visible, the visible face of Elixir Portugal, and in turn of Portugal to the Elixir, working with bioinformaticians, life scientists, and so on, using large amounts of data. These groups should be long-term, not limited in temporal scope, and should be subject to regular review. For instance, when we had our first assessment of the SAB, the existing communities, in particular the plant community, was assessed, and also at the Elixir level, when the nodes are reviewed, also the national communities are reviewed as well. And this should have strategic importance for the future of biodata PT and have broad scientific importance and relevance, and their membership must be drawn permanently from within the nodes and must be led from within Biodata. The benefits are the network itself, where you can collaborate, and also the events, the collaborative events that we organize. Also the communication, outreach, and branding, so we can provide visibility to the communities and their participants. Again, we can provide scientific support. We have a technical coordinator. He's Jorge Oliveira, and he's present here with us today. So if you want, you can address him directly. As I already mentioned, it benefits from SAB review and impact assessment. And we can provide, from Biodata Hub, we can provide additional support like shared folders, Zoom, and a community's calendar, Slack, all these tools that are necessary to facilitate the interaction amongst members. Regarding the contributions and responsibilities that we expect from communities, focus groups, and platforms, we expect them to have about 3 calls per year with agenda and meeting minutes that are available to all members upon each call. We have this annual meeting that is our biodata all hands. You can supply content to maintain the website up to date. We expect these groups also to continuing the effort to work in these topics that are defined by members and identifying these gaps that I previously mentioned, and that these are informed to biodata and shared with other structures. Like, for instance, if you are a plant community, you can ask from the support of the interoperability community to help you with the standards and your data management. Or if you need computing resources, you can interact with the computing platform. We also expect that there is a co-lead in these groups that shares the updates in frequent biodata meetings. We also encouraged everyone to attend the all hands, at least one representative per community, to contribute also to the national all hands and to the biodata talks program, as well as participating in grant proposals. We have selected here 3 examples of community services and platform services. For instance, some of you might already know the Ready for Biodata Management course that is provided by the training platform, and that it has been delivered already from north to south of Portugal with more than 300 participants. This number is already outdated. We also have a computing service that is delivered by the compute platform that has 3 nodes in Portugal, in Sicimar, in Gulbenkian, and in Técnico. It provides resources for many Portuguese organizations, being the providers of their own institutions, as mostly happens with Sicimar and Gulbenkian, or like Técnico, that provides machines to others, to other organizations, sometimes their own machines, sometimes providing support in the configuration of machines provided by INCD. Another example is a community service from the plant sciences community, and this is the CorCokeDB portal that you probably have heard of. And this portal aggregates tools and data and information from CorCokeDB research. So if you are doing research in this area, you can use this as a tool to improve your research efforts. And thank you very much for listening. And I now will give the floor to, I think, Celia. I kindly ask Sophia to present her, please.
Anna, I can present Celia, thank you.
Thank you.
I have everything. Okay, thank you very much for your presentation, Anna, for the great overview of what biodata is and where we're going with it. And for our next talk, I would like to present the 2 speakers, Celia Miguel and Danielle Faria. Celia is an assistant professor at the Faculty of Sciences at the University of Lisbon, and there she leads the Forest Genomics and Molecular Genetics Lab, and she's also the coordinator of the plant sciences community at biodata.pt, which is one of our most successful efforts. Her research interests are focused on genomics and molecular genetics of industrially relevant forestry traits. And Danielle is a senior researcher at LASIG, and he's also a member of Biodata and Elixir Portugal. He was actually the first person hired to work for Elixir Portugal back in 2015 and has been involved in a number of Elixir projects since. His research interests are centered on knowledge engineering, focusing on the biomedical domain. And without further ado, thank you very much, Celia and Danielle.
Okay, Katja, thank you. And hi, everyone. Thank you for the presentation. So we were asked a very difficult task to summarize in 10 minutes what has been our work in the context of biodata. So we'll try in this very brief presentation on the plant sciences research and interoperability to outline here what has been our work in the context of biodata. And in my perspective as a plant biologist, I will try to explain to you the reasons why it is so important, and I would say even crucial, to be able to share data so that the community of the plant researchers are able to share data in a fair way. And this means that data that we generate should be findable, accessible, interoperable, and reusable according to these FAIR principles that my colleague Danielle will then detail. I will try first to summarize the arguments and the challenges that we face in order to achieve these fair data in the plant sciences. And then Danielle will explain the tools and the resources that have been developed. And you will show very briefly also two examples of how these tools and these resources can be applied in the industry. So when this plant sciences community became active in the frame of the Elixir, so during the implementation phase of Elixir, our goal, and we were working together with a group of other researchers, our goal was to support the publication and access to plant phenotypic and genotypic data. And this data should be annotated according to well-established and accepted standards. And this is critical. So this publication of data in this way is critical for addressing some of the challenges that we face as a society and that are closely associated with the plant sciences, that are, for instance, the sustainable supply of food and other plant-derived materials, the establishment of a competitive life sciences sector, and ensure at the same time the environmental protection. Sorry. Yeah, okay. So to enable and to address these challenges, it is very important that we significantly advance plant breeding technologies. And this is very challenging because, I don't know, for those that are not familiar with plant breeding, this relies on the interaction of the genotype of plants with the environment. That will express, this interaction will express a number of traits in the phenotype that include, for instance, drought tolerance or increased yield or resistance to pathogens, among other targets of breeding. So in order to achieve this in a very efficient way, we should be able to integrate the wealth of genomic data that is available nowadays with plant phenotyping data. And so this systematic study of phenotypes on a very large scale and integration with genomic information will enable an efficient and more quick process of breeding, because traditionally breeding is a very slow process, especially for trees, for instance. But nowadays we have tools that allow us to accelerate this breeding. So, but these tools imply that we are able to use the data. And in recent years, very large amounts of data have been generated and released. and you all know about the new plant genomes that have been sequenced and that are available to everyone. And also a number of phenotyping platforms that are generating massive amounts of data every day. But while at the same time, this is a very good opportunity to advance breeding, it is also a really big challenge to be able to integrate these phenotypic data with the genome sequences, because there are still a number of gaps that prevent us to make these associations with, between the genotype and the phenotype in order to release tools for the breeders. And why is that? So what are these gaps that prevent this acceleration of breeding that are associated with the data? So these problems are that, for those that are not familiar with plant research, is that phenotypic data in plants are very heterogeneous, like in other organisms, but even more in plants, because we have to analyze phenotypes in different experimental settings, in the field, in the greenhouse. We generate different types of data, not only molecular data, but all kinds of descriptions of the plant's behavior during growth. And so these are very complex sets of data. Usually they are dispersed, so there is no central archive that you can go to and retrieve this data. Unlike, for instance, the DNA archives or protein archives that are curated and that everyone can access and understand. In addition, when these data are available, they are usually not properly annotated, because not all people use the same standards to describe the data. So the question here is to how to make this data fair, according to those principles that are mentioned in the beginning. And these should allow us to enable knowledge discovery and capitalize on the data that the community generates. So not only on our data, but only on the data that others in the community generate, because we can extract more knowledge if we integrate different types of data. So with this in mind, we in the plant sciences community worked in these past years to develop standards and ontologies to enable these fair plant phenotyping data, to implement repositories for these kind of data and user-friendly interfaces for data deposition and retrieval. But we have also worked to annotate and provide curated plant data sets and tools for plant data analysis, such as the one that Anna referred, the CorCookDB. And also one of our objectives is that the community adopts these principles. So we have been also providing training and having projects with the industry to exchange and apply this knowledge. So I will now hand over to Danielle.
Thank you, Sylvia. Share. So when we started working as a community back in 2015, I think, the landscape for fair plant phenotyping data was like this. So we have already an initial version of a standard for metadata, which was MyAPI. We had a standard for sharing and querying data, which was BRAPI. And there were ontologies and there were machine readable data publication formats such as ISA tools. But the connection between these things was very much lacking, and the degree to which we were satisfied with many of these things was also somewhat lacking. And so we set out to essentially create a unified landscape for publishing fair plant phenotyping data, because I mean that's always the problem with standards, is that there are too many of them, and so we we set out to make a single integrated, let's say, super standard out of these four. And so when we picked up MyAPI, first we realized that one of the goals of the Portuguese plant community was to focus on woody plants, and MyAPI was only focused on crop plants. So it was not really adequate for woody and especially forest species. And so we had to extend the scope of the standard to cover these species. And we also realized that there was no clear data model underlying the standard. So it was essentially just a list of fields and things you could provide, but the organization of those fields was really lacking. And so one of the main tasks we did was to create a data model specification that was in particular matching the ISA tab specification. So we would have interoperability with ISA tools. We added explicit ontology recommendations, again, to enable interoperability with the crop ontology, for example, and other plant ontologies. And we created an extensive mapping to the concepts that were in BRAPI, so that we could then actually use BRAPI to query MyAPI datasets. In addition, we created a number of machine-readable implementations of MyAPI, so an ISA tab template that was revised, an OWL version based on an ontology model of MyAPI, and templates for existing data management platforms such as FairdamSeek and Dataverse. With regard to BRAPI, then the problem was doing the reverse of what we did to MyAPI. So we actually had to map also BRAPI to MyAPI, make sure that the calls that are specified by BRAPI fully map to the mandatory concepts of MyAPI. And so that was the update that we did. We also ensured that we could have machine readable outputs resulting from BRAPI queries. So we created BRAPI to RDF and to JSON LD export functionalities, as well as mapping to ISA tab that allows both exporting and importing data to BRAPI. Additionally, as the Portuguese node of Elixir, we had the task of implementing a local BRAPI endpoint, which we did. So originally we implemented a BRAPI endpoint based on an SQL database, because we originally did not have a lot of data to work with. But as BRAPI was extended and we started having more data to integrate, we realized that we probably would be better off having an RDF-based solution using the MyAPI ontology as the underlying model. And so we're in the process now of converting our BRAPI endpoint to a triple store that is modeled using the MyAPI ontology. And so our BRAPI endpoint is part of a federated search service that is provided by the French node, and in which we collaborated also. That enables plant data to be queried across all Elixir nodes that implement BRAPI. More recently, we worked on enabling interoperability between bioschemas, which are an extension of schema.org for the life sciences. So schema.org is the markup that websites have that enable you to see nice vignettes when you do Google queries. So if you query for a movie, you'll see a little vignette about the movie. And so bioschemas essentially extend that markup for the life sciences. And we're interested in enabling pages, for example, describing a gene, or pages describing a plant species to actually be findable and machine readable in the same way. And so we are working on mapping bioschemas to the upper layers of MyAPI, so the fields that are useful for findability on the web. And we already did the markup of the Quark-Oak database pages with bioschemas, so all the genes and all the transcripts are tagged with adequate bioschemas markup. And we are also going to set up the Fair Discovery data center, a data service of the French node, to actually ingest bioschemas for resources that are not BRAPI. So having all these resources put together into a single cohesive landscape, we now started turning towards applying the things we had to concrete applications, namely in the industry. And so we, by happenstance, we came across during a training event a person that worked for the Phenospec's company. And this is a high-tech company that provides state-of-the-art plant phenotyping solutions. So with automated rigs and data capturing and an information system. And the person was actually the tech lead of the company, and he was very interested in implementing our standards, ensuring that their platforms actually capture data in a fair, compliant way, capturing all the fields that are mandatory for MyAPI, and that they could export their data via BRAPI calls so that their customers could actually query their data or easily integrate their data into their local BRAPI endpoints of the universities or the institutes where they're working, because a lot of the customers of this company are academia. And so this is, for us, a perfect marriage, because this enabled us essentially to have an automated solution that would facilitate the task that is quite burdensome for the end user of doing the annotation and description of the data sets in a fair, compliant way. So essentially, this tool will do a large part of the work for the user. A different collaboration we had with a different perspective, but also a critical perspective, was a collaboration with the Navigator company. And so the interest was more convincing them of the merits of data sharing and structuring data in a fair, compliant way. And they were actually interested in the idea of sharing some of the massive amount of data they have on eucalyptus breeding and see what we could do with it. So they gave us some pilot data sets, and they were interested in seeing how we could model those data sets semantically in our BRAPI endpoint that is now going to be shifted towards an RDF model. And so our goal is to showcase the data integration that we can achieve through that kind of modeling, and so demonstrate the value of verifying data, which would ideally lead them to share more data and lead other companies to share data with us. So I'll pass on back to Celia for the conclusions and wrap up.
Okay, can you?
Sorry, I can.
Okay, maybe it's not necessary for me to share. Can you go to the next slide? Yes. So just to conclude very briefly, we can say that the activities that we developed allowed to greatly improve this MyAPI BRAPI ecosystem, and it's now easier to enable fair plant phenotypic data publication by plant researchers. However, we are aware and recognize that these can still be improved, this process, to further minimize the amount of work that is required by the researchers to make this type of application through these technological solutions that are being developed with the Phenospecs that Danielle mentioned. But the main challenge here, and I'm sure that you all agree with this, is how to get people to be aware that the benefits of publishing data in this fair manner largely pay off for this effort that they do when publishing data. So the collaboration with the Navigator aims precisely to showcase the benefits of doing this, because they have a lot of data from many years, and they cannot take the most of these data because they are not able to integrate them easily with genomic data. So just to finish, I would like to acknowledge our collaborators. As I mentioned in the beginning, we have done most of these in collaboration with other Elixir nodes, with people in other countries, but also with the team here in Portugal from school, from ITQB Nova. I will not mention all the names that you can see here in the slides, and also our past collaborators, and those from the Elixir France, Netherlands, and Belgium that were instrumental to allow these advances, and others that are here mentioned. And thank you. So I think we have gone over time.
Thank you
Sorry
very
for that.
much, Celia and Danielle, for this very well-structured overview of the plant sciences community work over the years. Since we are running a little bit late and our Q&A session is only after the following presentation, I ask everyone who has questions for Celia and Danielle to please write them down if possible, because we will have an open Q&A after the next presenter. And I'm going to now just present our next speaker, Marta Silva. She is currently pursuing a PhD in computer science at LaSige at the Faculty of Sciences at the University of Lisbon. And she's also a researcher in the CATI H2020 project, where she's developing an integrated knowledge graph for artificial intelligence in personalized oncology. And she was also part of the biodata family. She was part of the development team of the Quark ODB portal before making the move towards the PhD. So Marta, thank you very much. You can share your screen and the floor is yours. Thank you.
Thank you. So if everyone can see fine and hear me fine, I will start. Today I will be talking about building a fair knowledge graph for personalized oncology. And this work is being developed in the context of a European project called CATI that aims to build a personalized medicine system empowered by artificial intelligence for renal cancer specifically. And personalized medicine is a paradigm that promises to deliver tailored therapy to patients in accordance with their molecular biology. And it is particularly necessary for oncology because each cancer ends up being genetically unique. So understanding the cancer and the patients at a molecular level will allow us to figure out which drugs or therapies will likely be effective, and then fine-tune their dosage. However, there are many layers of molecular and clinical data that need to be explored holistically and then integrated with existing knowledge in order to accurately predict the best therapy for each patient and each cancer. And this prediction can be made using sophisticated artificial intelligence methods that are able to take in all of these types of data and then output a prediction. However, one of the issues that we have is that these most sophisticated AI algorithms are black box solutions where we don't know the reasons that are behind the output of the algorithm. So for personalized medicine, what we need is explainable AI, which is to say we need to be able to tell the doctor that is using the system why a given prediction was made, because in healthcare we are talking about people's lives, so it's important. But to enable these explanations, what we need is shared context. In much the same way that we need shared context when we are understanding each other as humans. So we need to integrate the data that is fed into the AI with background knowledge in such a way that we can infer the causality of the AI predictions and then generate explanations that we can then show the user. However, building these semantic explanations comes with its own set of challenges, such as how do we impart the input data and the outcomes from the system with their meaning? How do we connect the data with this added meaning to what we already know? And then finally, how do we use all of this to derive effective explanations? And the solution can pass through the use of ontologies. Now, ontologies are knowledge structures that describe and semantically characterize the concepts of a given domain and the relations between them. So in the biomedical domain, we actually have hundreds of publicly available ontologies, each covering one or more subdomains. And as a whole, they provide excellent coverage of current biomedical knowledge. So ontologies are published in a standard machine-readable format, and they have external documentation about their intended scope and usage. But because they encode the knowledge in a structured and objective manner, they can also be reused for purposes that are beyond their original intent. Moreover, ontologies are not only fair in and of themselves, but they are also a cornerstone of fair data. They provide the means to describe data in a manner that is machine readable and objective, thereby enabling its interoperability, interpretability, and reusability. And we have a knowledge graph when data are described and integrated with an ontology in a single knowledge structure. So the ontology defines the semantic model, which is then instantiated by the data. So the knowledge graph ends up being one of the fairest ways to represent and document data. But how can the knowledge graph support explainable AI? So explainable AI techniques can be categorized according to their support of human reasoning. So they can be categorized into inductive reasoning, querying, and similarity modeling. And the knowledge graph is capable of supporting all three of these. So it defines axioms that support reasoning, it supports querying through Sparkle, and it enables the calculation of semantic similarity between entities. So for the process specifically of building a knowledge graph for the CATI project, we will be relying on integrating existing biomedical ontologies and then using them to describe the CATI datasets. We need multiple ontologies to capture all the subdomains that are relevant for personalized oncology, and developing a new ontology from scratch would not only be cumbersome, but it would probably fail to capture all the existing background knowledge that we have nowadays. So in the process, we start by identifying the relevant biomedical ontologies and gathering the biomedical datasets. We then perform a ontology matching step to combine the ontologies in a single holistic ontological model. And then we perform semantic data annotation to integrate the datasets into the ontological model that we built previously. And this will result in a knowledge graph, which can then be used as input for the AI system and will provide the much needed shared context that is required to develop explanations for the system's predictions. Now, when it comes to ontology matching, this is a process that establishes correspondences between the entities that are within each ontology. They can be simple correspondences where one entity in one ontology is equivalent to another entity in the other ontology, but it can also be complex, where the correspondences involve semantic expressions that contain one or more entities. So ontology matching is typically done between ontology pairs, but in CATI we will be matching several ontologies, which ends up increasing the complexity of this process. Moreover, we will be integrating the ontologies using these correspondences. So we have developed a method where we first start by clustering ontologies according to their semantic similarity, and then within each of these clusters, we perform matching, more sophisticated algorithms of matching just within this cluster. So we will tackle in this way the challenge of the scalability and the complexity of this part of the process. And when it comes to the data integration, we have to parse the data, of course, and establish the correspondences between the columns or keys in the data and the entities in the ontology. So we have to instantiate them. And we also have to capture the relations between instances and between each instance and its various data values. And of course, different types of data will require their own parsing and integration algorithms. So by having a comprehensive knowledge graph, instead of just having the direct information about our patient, what we have is a network of interconnected knowledge that can enrich the outcomes of the AI system and support our explanations. So if by inductive reasoning, we know that a patient with a mutation in the MET gene that increases the activity of tyrosine kinase is likely to have a good response to sunitinib, a specific drug that in turn inhibits tyrosine kinase. The question is whether we can establish this line of reasoning in the knowledge graph. And the answer is that yes, we can. We have a path within the knowledge graph that interconnects all of these little steps along the way and ends up connecting the patient to the drug. And what about similarity modeling? Can we find a patient within our available information, a past patient that responded well to sunitinib, and can we figure out whether that patient is similar to ours or not? And if we have that information in the knowledge graph about past patients, then yes, we can find out how similar those past patients are to ours, and then predict the most effective drug for our patients according to the similarity to the other patients. So in conclusion, we know that ontologies are able to support semantic explanations. We know that they increase interoperability and reusability of data, while themselves also being fair. It is possible to build a knowledge graph for personalized oncology using available fair resources. And this knowledge graph that is built with fair resources will itself be fair and also be able to support explainability in AI systems, which is very relevant in the biomedical domain. And that is it for my presentation. Thank you so much for your attention.
Thank you very much, Marta. So we can now open our Q&A session, and I invite anyone who would like to pose a question to any of our speakers, so Anna, Celia, Danielle, and Marta, to please raise your hand if you would like to ask a question, and I will, or actually Sophia, who is host of the session, will handle the process. And while you gather your thoughts and think of a question to ask our speakers, I do have a question, which is probably mostly directed to Celia and Miguel. Because at the end of your presentation, Celia, you mentioned the payoffs of FAIR. So how can we show others and motivate others that the investment on sharing and preparing the data according to FAIR principles gives a payoff in the future? And so maybe I would like to take this opportunity to ask you to tell us a little bit more about your experience of the payoffs. And so thank you, Celia.
First of all, I should say that all these resources that mainly Danielle showed have been available not for that long. So these have been developed in the last few years. But it is clear to me that if you are able to, because not every researcher has the resources to generate all types of data in their favorite system, for instance, in my favorite plant species. And so it is difficult for one research group to generate all the data that will give new insights in every aspect of the research. And so I, as a researcher, would like to, for instance, to have access to data that can be integrated with my data that are also available. And from that integration, we can draw new conclusions that cannot be taken or cannot be taken from the data alone from two research groups, for instance. I don't know if I'm explaining me very well, but for instance, the Core Code DB is an example because it resulted from the effort of a lot of research groups that established a consortium back in 2014. And so all these resources that were given by the National Foundation for Research were used by different groups. And now, together with the Genome Sequence Project of the Core Code, these resources are available for anyone that has the interest in Core Code to go to that portal and be able to search and to analyze data and to see everything that is available. So I think that is perhaps the best example at this stage that shows that all the community benefits from the effort of the others that have put their data available. And this is an example. Of course, these are sequencing data, so the sequencing data can be available in the archives, but the portal provides much more than that than just a sequence. So it provides tools for analysis. So for me, this is perhaps a good example. I don't know, Danielle, if you want to add another type of example.
Would you like to comment, Danielle?
I mean, I'm not really into data production myself, so I guess for me the immediate advantages in first-hand experience are less clear. But when I teach data management courses, I always ask people if they can reuse their own data that they produced a few years back, and that I think is one of the key advantages of actually putting in the work and going through the process of verifying your data and making sure that it's well documented, is that even yourself, you will be able to use the data generated years ago, or that your PhD student generated and has since left your lab. So normally when that happens, no one can resume their work or pick it up. So those are the kinds of applications that I think that resonate more with people.
Okay, thank you. Anyone from the audience would like to jump in and ask a question or comment on the discussion we've been having? We are not that many, so if you prefer, you can unmute and speak up. That would be fine as well. That's possible. Okay, so I actually have one more question, and this question is for Marta. So Marta, you told us about a project, a recent project. So for full disclosure, I am the local PI of the project that Marta is working on, and so this question has has a history or a background, but it was not rehearsed. So Marta, my question is, so we saw this perspective of the plant sciences community and of the work they did for the verification of data. But you yourself, you're not working so much on verifying data, but rather reaping the benefits of others that share their data in a way that made reusability and reproducibility possible. So can you tell us a little bit more about maybe the scale of the data and of the problem that you're tackling, so that people can see a little bit of the other side of the payoffs of FAIR?
Yes, I am the titular reaper of benefits. I am using the data that everyone else decided to make fair. So I don't know if it was 100% clear through my presentation, but so far everything that we have used at the point of the process that we are at has been ontologies that are available. So far we're working with resources that other people decided to make available, write documentation on, format in the standard manner, and I can tell you that it's fantastic to reap all of these benefits, because it's from the perspective of this work, we had, as I said, hundreds of biomedical ontologies to choose from, meaning that every single team of developers that created that ontology, that put work into it, that designed the model, and then put data into it and all that, they decided that they would share all of that with everyone else. And that's just, well, that's sort of the basis of science, isn't it? It's figuring out something cool and then sharing it with everybody. And we've been able to create an image, a full image of the biomedical domain using just these little parts that people have shared with everyone. So I can guarantee you that if you make your resources, your data, whatever it is you decide to share fair, if you decide to make it available for everybody in a way that can truly be reusable for whoever comes next, it can truly advance what everybody's doing, and we can do new stuff with old stuff. So it's great.
Okay, thank you, Marta. I see a question from Anna.
Thank you, Katia. I have a question, maybe for Marta, maybe for all. Precision medicine is expensive. And one of the main questions is how generalized can that be? Because not everyone would be able to pay. So my question is, how can these tools that take advantage of all available information minimize these costs and make precision medicine more accessible to everyone?
Well, it minimizes the cost because it's free. So if I want, for example, if I'm focused on renal cancer, I can go to Bioportal, which is a repository of biomedical ontologies, and I can pick and choose which ontologies I want to incorporate in my personalized medicine solution, whatever that may be. And that means that you don't have to start from zero. You don't have to spend time and money, which is oftentimes the worst part of it, in the beginning of just curating, selecting, making a model, figuring out how everything fits. And if you're starting from a sort of a further point down the line where you already have the knowledge that is organized, that is interconnected, and that you can connect it further by using more ontologies, then you sort of cut down on that very first step that is oftentimes harder to surpass. And are we gonna have free personalized medicine next month? Probably not. But
maybe I can comment, Marta.
Yeah, sure.
I would like to comment as well.
Okay, Danielle, go ahead.
So the main cost in personalized medicine still lies in actually generating the data. Okay, so it's still the sequencing, the molecular data generation that's the main cost. So I don't think that that the availability of fair data is that much of a saving in terms of the cost, but the approach that we are developing in CATIA, I think what it does is bring the most bang for your buck. So it ensures that if you put in the money to get your cancer sequenced and do the molecular studies necessary to get the data, then you can get the most effective and the most accurate predictions and treatment recommendations.
So maybe I can give you just a very practical example. So imagine that there is this one study that did all the work of sequencing patient data and getting transcriptomics or even proteomics data out of patients, and to compare patients that were taking a drug against patients that were not taking that drug. So you have that one study. Then you have another study that has another drug. And then you have another study that has another drug. If you want to build a system that actually compares all three drugs, okay, it is possible to reuse the data from these three different studies. Okay, so that is one of the things that having your data publicly shared and standardized and accessible and findable and interoperable and reusable, so fair, actually enables. So, and that is something that is being done in the project that we are working on. So we have data from a number of different studies that were made with slightly different goals. And we are analyzing those massive amounts of data under a common semantic framework that Marta is building that allows us to have this big holistic picture of the data that was actually produced siloed and separately. So that is one of the big contributions that fair data can give in this sense, is that data that was produced with one use in mind can serve other purposes later on and not just that initial purpose.
Thank you.
Okay, anyone would like to ask